In the present chapter, we will first understand about the extensive membrane network within the cytoplasm. When we see the cell under light microscope, it appears relatively devoid of any internal structure. It wasn't until the development of electron microscope in 1940s that the biologists began to note the diverse array of membranous structures present in the cytoplasm of most eukaryotic cells. It became evident from early electron microscopic studies followed by the biochemical investigations that the cytoplasm was subdivided into a variety of distinct compartments bounded by a membranous barriers. Although the electron microscope gives the detailed structure of cytoplasm membrane system, these pictures do not inform much about their functional relation to the structure. Thus, to understand and have an insight of the functionality of these membrane system, we must attempt to put the cell organelles into action in our own imagination. We have earlier studied about how the protein is synthesized using transcriptional mRNA from template DNA, followed by processing and translocation of mRNA from nucleus to cytoplasm, and finally translation of mRNA to polypeptide by ribosomes. The protein specifically meant for certain organelles, integral membrane protein and secretory protein, contain a special signal sequence at their N-terminus. This N-terminus signal of protein triggers the attachment of ribosome to an endoplasmic reticulum membrane and the movement of the nascent polypeptide into the lumen of endoplasmic reticulum. The signal sequence includes a stretch of 6 to 20 non-polar amino acid residues recognized by signal recognition particle or SRP. The SRP signal sequence complex of the nascent polypeptide is recognized by the SRP receptor located on the cytosolic surface of endoplasmic reticulum. This binding allows the polypeptide to elongate and enter into the lumen of endoplasmic reticulum. Finally, there is translocation of newly synthesized polypeptide for its long path towards its destination through vesicular trafficking. Now we turn our attention towards the movement of the nascent polypeptide to their destination by vesicular traffic through secretory pathway. To understand vesicular traffic, there are several techniques by which labeled proteins can be followed throughout the secretory pathway. Let us see few techniques regarding this aspect. Number 1 is pulse chase labeling with radioactive amino acids and with autoradiography technique. We can track the pathway of protein movement in different compartments within a cell. The second one is transport of fluorescently labeled protein along the secretory pathway can be observed by fluorescent microscopy. The third one is many of the components required for intracellular protein trafficking have been identified in yeast by analysis of the temperature sensitive secretion mutants which are defective for the secretion of proteins at the non-permissive temperature. The fourth one is cell-free assays for intercompartmental protein transport have allowed the biochemical dissection of individual steps of the secretory pathway. Let us be acquainted with vesicle and coat proteins. Small membrane bounded vesicles along with its coat proteins involved in transport of different proteins from one organelle to another are common elements in the secretory and endocytic pathways. These vesicle buds from the membrane of a particular parent or donor organelle and fuse with the membrane of specific target or destination organelle. Although each step in the secretory and endocytic pathways employs a different type of vesicle, studies employing genetic and biochemical techniques have revealed that each of the different vesicular transport steps is simply a variation of a common theme. So now we will see this common theme of vesicular transport. The bud formation from the donor organelle is initiated by polymerization of a set of soluble proteins known as coat proteins on the outer side of the vesicle to form a protein vesicle coat. These newly formed vesicle bud gather the appropriate protein which require transportation, also known as cargo proteins. Now the vesicle bud off from the parent organelle which contain cargo proteins, soluble coat protein, integral membrane proteins like membrane cargo proteins and membrane cargo receptor protein, the GTP binding protein and V snares. This V snares is a class of membrane protein which help the vesicle to find its destination and eventually help in fusion with the target organelle. Shortly after the vesicle is formed, coat of soluble protein is discarded and exposing the V snares protein on the exterior surface of the vesicle. Now the T snare protein is membrane protein located on the membrane of the target organelle. The specific joining of V snares to the T snares allows the vesicle to bring close physical proximity of the vesicle to organelle and allowing the two bilayers to fuse. Immediately after that, there is a discharge of the protein content into the lumen of target or destination organelle. Three types of coated vesicles have been characterized, each with a different type of protein coat. Now, each type of vesicle transports cargo protein from particular parent organelles to particular destination organelles. They are COP2 or cotomer associated protein 2. These vesicles basically transport the cargo proteins from rough endoplasmic reticulum to the Golgi complex. The second one is COP1 or cotomer associated protein 1. These vesicles mainly transport the cargo proteins into the retrograde or the backward direction between the Golgi cisternae as well as from the cis Golgi back to the rough ER. Clathrin vesicles generally transport the cargo proteins from the plasma membrane or the cell surface and the trans-Golgi network to the late endosomes. The same general scheme of vesicle budding applies to all these three known types of coated vesicles. Experiments with isolated or artificial membranes and purified coat proteins have shown that polymerization of the coat proteins onto the cytosolic phase of the parent membrane. This polymerization is necessary to produce the curvature of the membrane that is typical of a transport vesicle about 50 nanometer in diameter. Such structures, usually called vesicle buds, appear to be intermediates that are visible after the coat has begun to polymerize, but before the completed vesicle pinches off from the parent membrane. The polymerization of COPI, COPI2 and Clathrin coat proteins is regulated by DTP binding proteins known as ADP ribosylation factor, which is called ARF, and SAR1. ARF is present in the vesicle with COPI1 and Clathrin, while SAR1 is present in vesicle with COPI2 as coat proteins. It is believed that hydrolysis of ARF and SAR1 by GTP binding initiates the depolymerization of vesicle coat protein. After being released and shedding its coat, a vesicle fuses with its target membrane in a process that involves interaction of cognate snare protein. Now we will learn about the Clathrin coating and vesicle bud formation. Vesicle bud formed from plasma membrane and trans-Golgi network that are destined to form late endosome are coated with outer fibrous Clathrin protein and inner adapter proteins. Clathrin is so far the best characterized coat protein studied across the animal system. It structurally looks like three-limbed shape and called Triskelions from Greek meaning three-legged. Each limb contains one Clathrin heavy chain with 180,000 molecular weight and one Clathrin light chain having approximately 35,000 to 40,000 molecular weight. Triskelions polymerize to form a polygonal lattice with an intrinsic curvature. When Clathrin polymerizes on a donor membrane, it does so in association with adapter protein or AP complexes which assemble between the Clathrin lattice and the membrane. Each AP complex has 340,000 molecular weight. A specific association between the globular domain at the end of each Clathrin heavy chain in a Triskelion and the AP complex both promotes the co-assembly of the Clathrin Triskelions with AP complexes. This co-assembly adds the stability of the completed vesicle coat. Let us see about vesicle formation and target protein sorting. As described, Clathrin bind to AP complex to promote vesicle formation. So far, several distinct AP are recognized, for example, AP1, AP2, AP3, etc., and each related to specific vesicle formation and transportation of specified protein to different destination. By binding to the cytosolic phase of membrane proteins, adapter proteins determine which cargo proteins are specifically included in or excluded from a budding transport vesicle. Vesicles that bud from the trans-Golgi network reach to the lysosome by way of the late endosome have Clathrin coats associated with AP1. It recognizes a unique sorting sequence tyrosine XX5 where X is any amino acid and phi is an amino acid with a bulky hydrophobic side chain such as phenylalanine, leucine, isoleucine, etc. Some vesicles that bud from the trans-Golgi network have coats composed of AP3 complex. These vesicles mediate trafficking to the lysosome, but they appear to bypass the late endosome and fuse directly with the lysosomal membrane. At the later stage of vesicle formation, a cytosolic protein called dynamin is essential for release of complete vesicles. Once the vesicle is loaded with enough cargo protein to be transported, dynamine starts to collect at the neck of bud and polymerize using GTP, thus contracts the neck and aids in pinching off the bud from the parent membrane. Once the vesicle is formed, it needs to move towards the destination. So now we will learn about the movement of vesicle in the cytoplasm. For this, a dedicated protein known as kinesin is present in the cell. Kinesin 1 is a dimer of two heavy chains, each complex to a light chain with a total molecular weight of 380,000 Dalton. The molecule comprises a pair of large globular head domains connected by long central stalk to a pair of small globular tail domains which contain the light chains. Each domain carries out a particular function. That the head domain, which binds with microtubules and ATP, is responsible for the motor activity of kinesin, and the tail domain is responsible for binding to the membrane of vesicles, mostly through the kinesin light chain. Dimeric kinesin molecule exerts a force of 6 piconewtons, which is sufficient to pull a bound vesicle through the viscous cytoplasm. The kinesin step size is of 8 nanometer, that matches the distance between successive alpha or beta tubulin monomers in a protofilament, suggesting that kinesin binds to only one or the other monomer at a time. Researchers have established that a double-headed kinesin molecule moves along a single protofilament with one head always bound to the microtubule. As a result, a kinesin molecule can move along a microtubule for a long distance without detaching from it, a property referred to as processivity. Because of their high processivity, dimeric kinesins are very efficient in transporting vesicles from one part of the cell to the another. It has been discussed that the most of the sorting signals are a short stretch of amino acid sequence located within the protein, which are identified by the adapter proteins and accumulated in budding vesicle. Here we learn about a new sorting sequence made up with the carbohydrate residue known as mannose-6-phosphate or M6P signaling. This M6P is tagged to soluble lysosomal protein during the process when it passes through cisternae of endoplasmic reticulum. The addition and the initial processing of N-linked oligosaccharide precursors in the rough endoplasmic reticulum is the same for many lysosomal enzymes and the other secreted proteins, yielding core of 8 mannose and 2 N-acetyl glucosamine chain. In the cis Golgi, the N-linked oligosaccharides present on most lysosomal enzymes undergo a two-step reaction sequence that generates M6P residues. The addition of M6P residues to the oligosaccharide chains of soluble lysosomal enzymes prevents these proteins from undergoing any further processing. In trans-Golgi network, the transmembrane M6P receptor binds to the M6P residues of the lysosomal destined proteins. Once the tight binding of the M6P receptor to protein takes place, they accumulate in budding vesicle formed by Clathrin and AP1 coat at cytosolic phase. This vesicle buds off the trans-Golgi network and gets transported to endosome by kinesin complex, resulting into fusion with endosome. Binding of M6P receptor to M6P is possible only at the slightly acidic pH, that is approximately 6.5 of the trans-Golgi network, but not at pH less than 6. When the vesicle is fused with endosome, which has pH 5 to 5.5, the binding is detached, resulting into release of lysosomal protein and preventing reattachment of the protein to receptor. Furthermore, in late endosome, the phosphatase removes the phosphate group from the M6P, thus also preventing rebinding of M6P receptor. The free M6P receptor along with the membrane is recycled into the trans-Golgi network for fresh sorting of lysosomal enzymes from Golgi lumen. So far we have studied about the sorting of the protein and movement of these proteins through vesicle to plasma membrane or other organelles. But cells can also ingest material from the surrounding with the help of receptor mediated endocytosis process. It is seen in macrophages which internalize the bacteria after binding it with the receptor. During the whole process, the macrophages extend the cytoplasmic arms on either sides of the receptor bound and form a phagocytic vesicle. Similarly, small region of the plasma membrane invaginates to form a membrane limited vesicle about 0.05 to 0.1 micrometer in diameter. In one form of endocytosis called pinocytosis, small droplets of extracellular fluid and any material dissolved in it are non-specifically taken up by the cell. In general, eukaryotic cells internalize cholesterol or triglycerides containing particles called low density lipoprotein, which is called as LDL, the iron binding proteins, transferrin, intrinsic factor which carries vitamin B12, many protein hormones as for example insulin and certain glycoproteins by this process. Generally, transmembrane receptor proteins present in plasma membrane that function in the uptake of extracellular ligands. These extracellular ligands are internalized from the cell surface during endocytosis via Clathrin and AP2 coat formation on cytosolic phase. Such form pits further form small phagosome or pinosome that are destined to fuse with endosome or lysosome, and then the receptors are sorted and recycled back to the cell surface, much like the recycling of M6P receptors to the plasma membrane and trans-Golgis. Cells actually contain two functionally distinct classes of receptors, one for hormones and one for nutrients. Like the peptide hormones, the nutrient carriers are too big to pass through the plasma membrane and therefore they too must be internalized by endocytosis. Although there is some overlap in the way that each group is processed, there are some general differences that can be distinguished between the two. As for example, in nutrient processing pathway, the receptor usually pre-clustered, their internalization and recycling is continuous and they recycle by short loop pathway, whereas in hormone processing pathway, the receptor usually not pre-clustered but by diffuse, Their internalization is ligand-induced and discontinuous, as well as they recycle by long loop pathway. The two pathways also differ in their processing of the internalized ligand-receptor complex. In the nutrient pathway, the endocytotic vesicle called an endosome or receptosome becomes acidified via an ATP-dependent mechanism. At about the same time, slender tubules begin to emerge from the surface of the vesicle. This entire complex has been called curl, which is called as compartment for uncoupling receptors and ligands. The acidification disrupts ligand receptor binding and the unoccupied receptors migrate into the tubules while the free ligand remains in the vesicle. The tubules then detach and return to the plasma membrane where the receptors are recycled. Finally, the vesicle fuses with lysosomes and the ligand is degraded. This pathway is known as the short loop. Hormone receptor complexes are treated in a similar manner, except that the liberated receptors first go to the Golgi apparatus before returning to the plasma membrane. This is called long loop. At the end, in this lecture, we tried to trace the pathway of protein from its synthesis to sorting, as well as the movement of protein in different vesicles towards its destined place, like exterior to the cell, plasma membrane, organelles, etc. We saw vesicles are the carriers of the protein which are formed from the parent organelle and travel towards the target organelle or the exterior of the cells. Code proteins like COPI, COPI and Clathrin in association with adapter proteins and the other cytosolic proteins help in vesicular budding, sorting and accumulation of protein in the carrier vesicle. Clathrin along with APs recognize specific signal sequence of target protein and collect in vesicle which bud out by dynamic polymerization. These vesicles are then transported to different cytoplasmic location by kinesin, a motor protein associated with microtubules. Proteins are also sorted by carbohydrate moiety like M6P, which are recognized by transmembrane M6P receptor. M6P receptor binds only in pH range near to 6.5 and hence detached from the M6P after fusion of vesicle to endosome, where the pH is almost between 5 to 5.5 and recycle back to the parent organelle. Similarly, receptor mediated endocytosis also employ Clathrin or AP2 protein and forms a pit resulting into phagocytosis and pinocytosis. The pathway for receptor processing for nutrient and hormone are different. They are known as short loop and long loop pathway respectively.